So, what drug has saved the most lives throughout history? Now, you might think, oh, penicillin, an antibiotic, right? That's obvious. Not so. Vaccines as a drug class have saved more lives throughout history than any other drug class, 1 billion lives and counting. It's just totally incredible. I mean, it was 225 years ago that Jenner vaccinated somebody for smallpox, and now the disease is eradicated. However, we have not been so lucky with other diseases, such as HIV. For those of you who've had the COVID vaccine, we still walk around a little bit in terror, right? Because we don't know whether or not Omicron or some variant is going to come visit us. So what I'd like to propose to you today is that AI can make these vaccines better. So today we're going to embark upon a journey. Together we're going to design a new vaccine, a pan-variant COVID vaccine that can address some of these risks. One of the challenges with current vaccines, they're all based on spike. And you might think, oh, they're different, right? Because mRNA delivery or viral delivery or protein delivery, well, it's all the same team on different vehicles, right? And so what we need to do is to come up with a new payload strategy, a payload strategy based upon the insights that we have in this room, AI-based insights. Now, what's the problem with the current vaccines? That's escape, right? They run away from current therapeutics because the way vaccines work at present, the licensed vaccines, is to induce an antibody response against the face of the virus, and the face changes over time. And we actually don't know exactly where it's going to go in the future, nor is it really easy to generate a single antibody that protects against all possible future variants. So what's the alternative? I mean, how could we get around this fundamental challenge? Well, here's a proposal. We're not going to go after the face of the vaccine, we're going to go after its heart. We're going to go after the parts of the virus that can't change, that are mechanical, that make it work. Now, how could that be possible? Well, it turns out that every virally infected cell displays on its surface little bits of the virus that it contains. And so we're going to program our immune system to see those bits of virus and kill those cells. Now, this is a challenge because you have 6 genes, 3 from your mom and 3 from your dad, that encode for these little billboards that display the bits of virus. So each person displays a little bit different viral components. So somehow the vaccine we design has to take this into account. So our first use of AI, using new methods we developed, is to discover what bits of the virus, here shown on the y-axis, are going to induce an immune response in people with what's called an MHC allele on the x-axis, or a variant of these billboard molecules. And where there is white in the matrix indicates that there's going to be an immune reaction between that epitope and the allele that you have. So using this, what we would like to do is to program these killer T cells. Here's an example killer T cell. It's on patrol, and it's lethal, and it's dangerous, folks. It's out there looking for a target cell. And just to show you how dangerous this little puppy really is, let me show you the work, okay? Now, it's going to go find a blue cell that contains non-self proteins. Ooh, poor blue cell. So sad. It contains epitopes that are non-self, and the red cell expels little components that burrow and drill into its cell membrane, killing it. So how can we select a set of epitopes to program our immune system to see such cells? Well, we're going to use combinatorial optimization. Let's say we have 488 or so little epitopes that are invariant across the heart of COVID, and we have over 1 million combinations of billboard molecules. We want to select, say, 30 of these epitopes on the left-hand side to program our immune system to get maximum population coverage with the maximum number of hits per person. And when we look at this kind of approach, you can see the middle plot here, which is at least 5 hits per person. Other published designs are little red X's, and the blue line is the frontier of the MIT T-COVID vaccine. And you can see it's superior population coverage for that number of hits. But the question is, does it work, really? Now, how could we test this? Well, it turns out with influenza, you can actually pay somebody in the UK to infect people with influenza, but we can't do that with COVID. So what we have to do, we have to make the vaccine and test it in an animal model. So this is what the vaccine actually looks like. It's encoded in a polypeptide with signal sequences that are red and blue with the epitopes in boldface type. This is packaged using an mRNA LNP construct, same as the Pfizer COVID vaccine. And we're going to use it to immunize an animal that has part of a human immune system. We're going to immunize on day 0 and day 21, 3 weeks later, and then we're going to expose this mouse that's been immunized to beta variant of COVID, which is very pathogenic and has a host range that includes mice. So when we do this, we see on the top plot here that we get a good immune response. Our programming is successful. We're getting the killer's T cells to recognize bits of the virus in the mouse. On the lower left-hand side, what you can see is after exposure to the virus, 24% of the cells in the lung, the nucleated cells, are these killer T cells patrolling around looking for virally infected cells in the lung, whereas in an unimmunized animal, only 7% of the cells are killer T cells. And on the right-hand side, what you see is that animals that are not immunized, 80% of them die according to our criteria. However, immunized animals all survive with the MITT COVID vaccine as well as with the Pfizer-BioNTech control. So the thought I want to leave you with today is that AI provides novel medical solutions. It's just beginning, not only for vaccines, but as we also heard in the previous talk, for other kinds of therapeutic modalities. And so it's a very exciting time. Thank you very much.